Rahim. Welcome back, friends. Welcome to another video lecture by Biology. In a series of video lecture, we are going to talk about DNA replication in prokaryotes. So let me write DNA prokaryotes. Usually in a bacterial cell, or we can say in E. coli. E. coli. Okay. Now this is the first video lecture of the DNA replication, and in this first video lecture we are going to talk about different kinds of proteins and the stages which are involved in the DNA replication in case of the prokaryotic cell. So we are going to talk about different kinds of proteins. Almost there are 30 proteins which are involved in the DNA replication in case of the prokaryotic cell. And we have 3 stages which are involved in the DNA replication. These are the initiation, we have elongation, and termination. Okay, now this is the first video lecture, and in this first video lecture we are going to talk about proteins or the enzymes or the replication machinery. We also say that replication machinery which are involved in the DNA replication. So we can also say that this is actually the basics. This is the introduction of the DNA replication in prokaryotic cell. First of all, let me explain that what is actually DNA replication. DNA replication is that process in which a DNA molecule can produce 2 copies of the DNA, 2 copies of the daughter DNA. If let's suppose we have this strand of the DNA, okay. Now from this DNA strand we get 2 daughter DNA. By using this DNA as a template we get 2 daughter DNA. Now this is called replication. So it is the process, let me once again tell you, it is the process in which we get 2 daughter DNA from one parental DNA, or from old parental DNA we get 2 new DNA strand. We call that replication during cell division. Okay, and usually in this case the cell is divided through binary fission because it is a bacterial cell, so it is divided through a binary fission. Let me tell you a little bit about the binary fission, that how actually the cell are divided during binary fission. In this case, you know, a bacterial cell, we have a bacterial cell. So let's say this is a bacterial cell. It is ready to divide. So what actually happened? This bacterial cell are elongated. Now it is elongated. They have also their genetic material as well. Okay, whenever they are elongated, so their chromosomes will be also divided. So their chromosome will be also divided. So we get 2 chromosomes. Not 2 chromosomes means we get means another copy of the chromosomes. And due to this way we get 2 daughter cells having the bacterial chromosomes. Usually bacteria have a circular chromosomes. Okay, so how this binary fission is possible? How this process is possible? This process is possible by the DNA replication. So this chromosome is actually the DNA chromosomes. Whenever it is replicated, the cell is also replicated. And in this case, the cell is divided, not replicated. The cell is divided through the binary fission. Okay. So inshallah, in some other video lecture, we will explain the eukaryotic DNA replication. Now, DNA replication is different in case of bacteria, in case of eukaryotes, and in case of archaea. It is different. And it is usually different at initiation stage. Elongation and termination is the same in each of the cells, but the initiation is different. Although elongation and termination is also different, but a little bit. Now, what kind of the proteins which are involved in the DNA replication? Once you get the idea about the proteins which are involved in the DNA replication, then in the next video lecture, which we are going to talk about initiation, elongation, and termination, will be very easy for you. So we have different kinds of the protein. The first protein we call that DNA proteins. DNA proteins. We have three types of the DNA proteins. The first we have DNA A proteins, second we have DNA B, and then DNA C proteins. DNA A proteins are usually responsible in the initiation process. Initiation process. Okay, this DNA A protein can bind on the origin of replication, which we can say that ORI-C. Okay, then we have another DNA protein we call that DNA B protein. This DNA B protein are involved in to break the hydrogen bond. This DNA B protein are usually involved to break the hydrogen bond. They are acting as a helicase, which are usually found in the eukaryotic cell. So acting as a helicase and break hydrogen bond between strand. Between the two strand, they can break the hydrogen bond. Usually in 5' to 3' direction. Okay, let me once again tell you, they act as a helicase and they can break the hydrogen bond which are found in the two strand and separate them, and they can break the hydrogen bond in the direction of 5' to 3' direction. And moreover, this strand have a DNA primase activity. This strand have what? DNA primase activity. What does it mean? It means that they can also add a DNA nucleotide, or they can add a DNA primer which contain almost 10 nucleotide of the DNA. Okay, now then we have another DNA protein we call that DNA C protein. Now, what are the function of DNA C protein? They are involved to proceed the B protein in 5' to 3' direction. Okay, so they proceed or directs DNA B protein. Okay, DNA B protein just bind. DNA B protein just means, let's say here the DNA, let's say this is the DNA B protein. They just bind here. They can't go in forward direction, or they can't go in 5' to 3' direction. They will be proceed in 5' to 3' direction. When DNA C protein can bind here, then they can start their proceeding. Okay, this is the job of DNA C protein. Now we have another protein, DNA gyrase. DNA gyrase or DNA gyrase, we can say like that. It is usually involved to release the tension or to relax the tension. Relieves the tension. Or we can say, then we have another protein which are involved in DNA replication. We call that SSB protein. We also call that single stranded binding protein. Now this SSB protein are usually involved, they prevent the strand from binding. Let me once again tell you, let's suppose this is the DNA strand. We melt it, so the DNA will be separated from each other. Okay, whenever it's separated from each other, then there may be a chances that these DNA are again bind to each other, because the strand are complementary to each other. To prevent this phenomena, we use or there is another enzyme or protein used, we call it single stranded binding protein. They can just bind to the strand and prevent them for the binding. Okay, then we have another strand, we call that protein N single prime, N double prime, and protein 1. These kind of protein are usually involved in the assembly of the chromosomes. Now, what are the chromosomes? Chromosomes are the organelles. Chromosomes are the primer. These are the nucleotides. For example, just like that, we call that primer, which contain nucleotide sequences. We call that primer. Now, this is called as chromosomes also. So these chromosomes can also be synthesized by protein N prime, N double prime, and 1 protein. Then we have another protein which are involved. We call that DNA primase. These DNA primase are usually responsible in the synthesis of RNA primer. Synthesis of RNA primer. Okay, so they can synthesize the RNA primer. Then we have another proteins, we call that DNA polymerase. In short form, we can like pol. We have two types of the DNA polymerase which are involved in the DNA replication in prokaryotes. We have also DNA polymerase. We have also 3 types, but here we will discuss 2 types of DNA polymerase. We call that DNA polymerase 1 and DNA polymerase 3. So DNA polymerase 1, which are involved, they are involved to remove the RNA primer and fill the gap by adding a correct nucleotide, DNA nucleotide. For example, we have this strand. Now this is the RNA strand. How we can remove it? All of you better know RNA and DNA strand are same. They have same nucleotide, but just one nucleotide is different. In RNA, we have uracil, and in DNA, we have thymine instead of uracil. So what actually the DNA polymerase 1 can do here? DNA polymerase 1 can cleave the RNA primers, or they can cleave the uracil. And when they cleave the uracil, so we get a gap. So this gap is then filled by DNA nucleotide, which is the job of DNA polymerase 1. So it is performed by DNA polymerase 1. Then we have another DNA polymerase, we call it DNA polymerase 3. This DNA polymerase 3 can also involve to add the nucleotide in 5' to 3' direction, adding nucleotides in 5' to 3' direction, and they have also exonuclease activity. Exonuclease exonuclease activity. Exonuclease activity, or we call that proofreading. They have also proofreading. Now what does it mean? It means that whenever the new strands are formed, then they just check again the strand. Whether there is incorrect nucleotide are present, they cleave it and add the correct nucleotide. This is also the job of DNA polymerase 3. But they can start their activity when there is the RNA primer. Okay, they can't start their activity without the RNA primer. Now then we have another proteins. Okay, then we have another protein, we call it ribonuclease H. Ribonuclease H. Now what are the job of this enzyme? They can also remove the RNA primers. Remove RNA primers. Okay, then we have another enzymes. So let me erase this upper side and let me write some other proteins which are also involved in the DNA replication in prokaryotes. Then we have another DNA protein, we call that DNA ligase. We have another enzymes. This is actually DNA ligase enzyme. And all of you better know what are the job of DNA ligase. DNA ligase can just seal the nick of the DNA fragments. Seal DNA fragments. Seal the nick of DNA fragment. Okay, or they can just prepare the bond, phosphodiester bond. For example, let's say there here is the phosphodiester bond is missing, so they can just create the phosphodiester bond by joining the two DNA molecule, by joining the fragment of the DNA molecule. Okay, then we have another enzyme, we call that DNA replication termination site binding protein. Okay, so in short form we can write RTSBP. Now these kinds of proteins are especially involved in termination stage. In termination stage, the replication process is completed, and then the two stranded are divided. So they can, let's suppose if this is the bacterial chromosomes, which is the double stranded, and whenever they can produce another bacterial chromosomes, so this is the bacterial chromosome, or now we can get the bacterial chromosomes. Now the replication is completed. So how this chromosomes will be separated from each other? How this circular DNA will be separated? So it will be separated by using this DNA replication termination site binding protein. They just cleave it and separate these two DNA from each other. So after that we get the two circular DNA, and the replication is then completed. Okay, so involved in termination. Last we have another protein, we called it Topoisomerase. Topoisomerase enzyme 4. Now what are the job of topoisomerase enzyme 4? They can segregate the chromosomes. They are usually involved in the segregation of the chromosome, in the separation of the chromosome. Once the DNA are formed, let's suppose, then they can just involve to separate the DNA into two form. Okay, so that's the main important proteins which are usually involved in the DNA replication in case of the prokaryotes. Inshallah, in the next video lecture we are going to talk about initiation in a detail. Okay, and we are going to talk about the proteins which are involved in the initiation types. These all are the protein which are involved in initiation, elongation, and in termination. Okay, so stay tuned and continue to watch the video. Thank you so much.